Hello, my name is Tolulolawale, and I'll be making this presentation titled Bisphenol-A, Associated Epigenetic Changes in Young Adults in Northern Nigeria at the 13th International Conference on Environmental Mutagenesis in Ottawa, Canada. Endocrine disruptors are chemicals that mimic hormonal activity, interfering with the functioning of endocrine systems in living organisms, and they can be found in a variety of household products. A typical example would be bisphenol-A that is used in the manufacturing of polycarbonate plastics and the lining of cans. And there has been concerns over the potential health risk of bisphenol-A in consumer goods because bisphenol-A has a strong estrogenic effect and it can compete for the binding of receptors over other hormones. And studies have suggested that environmental exposure to bisphenol-A during critical windows of development is associated with epigenetic changes and reproductive abnormalities because the mechanism of action of bisphenol A is strongly linked to the HPG axis that regulates the release of hormones. And so with the unregulated use of BPA in consumer goods and the increased utilization of BPA containing products in Africa, and seeing that BPA is poorly monitored in Africa, research sought to investigate associations between BPA exposure, reproductive hormones, and DNA methylation in whole blood from young adults. Healthy Covenant University students within the ages of 18 to 24 years were recruited, and DNA was isolated from the whole blood samples that were collected, while urine was collected in BPA-free bottles. Hormonal and BPA assays were done using ELISA, and the DNA was later bisulfite converted and pyrosequenced to detect DNA methylations at OXA10, PPAR alpha, and estrogen receptor genes. BPA was detectable in all urine samples in the studied population, but though the range reported in our study was lower than what has been reported in Africa and across other continents, it might not be a true representative of BPA levels in Africa, since studies are limited on monitoring of BPA. In measuring the strength of relationship between BPA concentration, hormones, and DNA methylation, in males there was a strong negative correlation between BPA and PPAR alpha at CpG site 1. PPAR alpha is a steroid hormone receptor and it is inhibited by estrogen, and it acts by regulating the expression of several genes that are vital to lipid and lipoprotein metabolism. BPA is a known inducer of PPAR alpha expression, and it can go on to inhibit or lead to abnormal physiological functioning of sex steroids. In females, BPA exposure was associated with hypomethylation of OXA10 and estrogen receptor at CpG site 1. And alterations in OXA10 methylation has been associated with several human cancers. We also saw sex-specific differences in the adjusted models accounting for sex and BMI. Overall, BPA research in Africa is nascent, and there is need for more studies, especially biomonitoring studies, to determine the daily intakes of BPA in Africa and the toxicological impact of such exposures. These studies would help to regulate policies and also the use of BPA in Africa. Thank you very much for listening.